Alright, welcome to my channel. Today's topic is cleanroom classification in pharmaceuticals and cleanroom classification grade A, B, C, and D. And classification is also based upon ISO standard, that is 14644-1, classification of cleanroom by particle size. A little bit about me. So I have done bachelor's in pharmaceutical sciences, master's in pharmaceutical operations and management. I have over 16 years experience in quality assurance, working in the shop floor environment of pharmaceuticals, both sterile and non-sterile oncology products and terminally sterilized products. Uh, I have worked with drug products, drug substance, and sterile manufacturing such as ampoules, vials, lyophilized products, and pre-filled syringes, and combination products. During my tenure, I faced many audits at different levels. I have worked as a front room coordinator, worked as a scribe, and then gave tour to the auditors. Pretty much, every role I have faced in the audit. I've given a link to my LinkedIn connection here. If you would like to connect with me, I would appreciate that. I have worked in India, United States, and currently I'm working in Canada. This presentation would be helpful if you're working in pharma or med device or API manufacturing. If you're working in quality control, quality assurance, validation, manufacturing, or if you're working in a chemical manufacturing company as well. So, coming to the question, what is a clean room? Looking at the FDA guidance for an industry document, clean room is defined as a room that is designed to design, maintain, and control to prevent particle and microbial contamination in the drug products. Such a room is assigned and reproducibly meets an appropriate air cleanliness classification. There are two things mainly the contamination it talks about. One is contamination from particles, which is non-viable, basically non-living. And second thing is microbial contamination, which is viable, which is living. So microbial contamination could be from a bacteria or fungi. So what is an airlock? So airlock is a small room with interlocked doors constructed to maintain air pressure control between adjoining rooms, generally with a different air classification, air cleanliness standards. The intent of an aseptic processing airlock is to preclude ingress of particulate matter and microorganism contamination from a lesser controlled area. Parameters controlled and monitored in clean room are temperature is monitored and relative humidity is monitored and air velocity is monitored, not as frequent as temperature and humidity, but air velocity is done frequently as a part of room qualification or room qualification. And the same thing goes with air changes, which is done as a part of room qualification and room requalification. And airflow is also done during the qualification. Differential pressure is continuously monitored throughout the day. It could be a manual system, by recording what is the differential pressure, looking at the differential pressure gauge, or it can be done with a building monitoring system, which continuously monitors the differential pressure in the rooms. So why is a clean room needed? What is the purpose of the clean room? So the main purpose of the clean room is to prevent particle contamination in the product that is being manufactured. So whenever I say particle, I'm referring to a non-viable particle or a non-living particle throughout the presentation, unless I explicitly say it as a viable particle, viable contamination. So clean room is also needed to prevent microbial contamination in the product that is being manufactured. Let's understand what is the main source of contamination. So main source of contamination can come from many sources, but mainly it is from the people. People who are working in the clean room, like if the skin gets exposed, it could be from the skin, it could be from mucus, oil, hair, cosmetics used, cosmetics are technically not allowed in the clean rooms. We'll get to that more in the presentation, or maybe a different presentation. I'll exclusively make for clean room do's and don'ts. And from the microbes and from the clothing that a person wears, which is a gown. The people who are working in the clean room should wear a gown that is dedicated for the clean room. Or the contamination can come from the equipment that is used, like if you are using a depyrogenation tunnel, or if you are using a filling machine, or if you are using a vial capping machine. So it could come from that as well. So especially in the vial crimping area, it generates particles. So it could be due to machine wear and tear. And the third thing is contamination can come from the utilities that is used in the manufacturing, be it a compressed air, nitrogen, or argon gas that is used in the clean room. That is why in clean room, if any of these utilities are used, there is a filter that is a 0.2 micron filter that is typically used in the clean area to prevent the contamination. Contamination can come also from the materials or the components that is used in the manufacturing. So this is the classification of clean rooms, and most widely accepted standard is ISO standard, which is 14644, that defines clean room clean rooms are classified based upon the particle size concentration. So there are two things to remember here. One is size of the particle, and the second thing is number of particles that are in a cubic meter volume of air. The latest standard is issued in 2015, that is ISO 14644-1, issued in the year 2015. So ISO standard classifies room from ISO 1 to ISO 9. But in this slide, I just gave the information of, ISO 5, 7, and 8. As you can see here, what are the size of the particles and your maximum allowed particle concentration criteria per cubic meter of air. So we can see here, in ISO 5, the zero point 0.5 micron size particles should not exceed 3520, and in ISO 7, it is 352,000, 352,000 per 0.5 micron particles. Whereas 5 micron particles, should not exceed more than 2,930. ISO 5 is, higher classification in terms of cleanliness compared to ISO 8, which is a lower classification. As you can see, the particles are less per cubic meter in ISO 5 area compared to ISO 8 area. This is a classification based upon ISO standard. Prior to that, there is another standard, that is Federal Standard 209E, but after the ISO standard was issued, the federal standard has been superseded. People are no longer using that terminology of class 100, class 1000, or 10,000. But just for the sake of reference, it's always good to understand, if somebody says what is class 100. So, I mean, though it is not typically used, it's good to know. So class 100 is typically equivalent to ISO 5. It is equivalent to ISO 5. So an ISO 5 particle concentration is equal to class 100 and approximately equals to EU grade A, which I'll talk about in the next slides. So this is EUGMP NX1, and classification per EUGMP NX1 of the clean rooms is grade A, grade B, grade C, grade D. Grade A is a higher classification compared to other grades, as you can see here, B, C, and D. And here also you can see the maximum number of particles allowed per cubic meter of air, For example, as you can see, grade A at rest is 3520. There are 3520 particles, a load limit per cubic meter of air. And in operation also is 3520. So here one thing is important to know. So in EUGMP, it gives the limits at two different levels. One is at rest and in operation. At rest means, let's say the filling machine is there in the filling area, but filling machine is not in operation. It is just there. The facility is built and facility is qualified, and with all the equipment inside, but there are no people. So that state is called at rest. So in operation is similar to at rest with an exemption of filling machine is there, but the filling machine is in operation, like it is actually filling some product. So that is in operation meaning. Right. So in case of grade A, the limits are same for at rest and in operation. Whereas for grade B, as you can see here, at rest the limits for 0.5 micron count is 3520, whereas for grade B in operation, the zero point 0.5 micron count a load limit is 350 to 1000. This is just an example here. Okay, so per FDA guideline for industry, sterile drugs produced by aseptic processing issued in the year 2004, these are the particle count limits and these are the microbial count limits. Though you see the terminology here as class 100, and you always refer to the standard. So there is one thing important to mention here. Um, the FDA is a guidance, right? And whereas ISO is a standard, there is a big difference between a guidance and a standard. Guidance is basically, it guides this is how it needs to be, whereas standard is something you must follow. There are no exceptions to it. So the same thing here, grade A or ISO 5 limits are same in both at rest and in operation. So what are the applications of pharmaceutical operations by clean room by grade? So what pharmaceutical operations are conducted in grade A or ISO 5? So these are some operations conducted in ISO 5 grade A. This is a aseptic area filling zone where the product is exposed. Example, machine assembly, filling machine assembly, or while filling and while stopping area, or autoclave unloading area under laminar airflow, or ampule filling area where aseptically filled, where the product is aseptically filled, and pre-filled syringe filling area and lyophilizer loading area. And second thing is ISO 5 or ISO 7. So here why I have written ISO 5, ISO 7 is grade B, which is like I grade B at rest, and in operation the limits are grade C. So grade B is a supportive area for grade A. So typically in aseptic manufacturing area, the filling room is grade B, and the filling area within a CRAB you say, or within an isolator or within a laminar airflow, under the laminar airflow or inside the isolator or inside the CRAB, which is a closed restricted access barrier, that classification is grade A. But the filling room supporting area, or where the Where the filling machine is, that room is typically grade B. Or the vial filling room outside lab, a room in which filling machine is installed, or a supportive room for autoclave unloading area. Supporting room for a lyophilizer loading or unloading area. That is a grade B area. And grade C is filling area for a terminally sterilized products. When you say terminally sterilized products, so after the filling is completed, that is sterilized by autoclave. The finished product or a drug product is sterilized by autoclave. So those are terminally sterilized products. Filling area of terminally sterilized product can be in a grade C, and compounding area where the drug product is manufactured, that is grade C, and solution mix rooms or component preparation rooms where the components that is needed for filling the product, like filling needles or the stopper valves. So those components are washed and then packed into the Tyvek pouches before sterilization. That component preparation area needs to be grade C or ISO 7, or ampule washing area or a vial washing area, or garment washing areas like garment washing area for sterile area garments. So those are some areas where which requires grade C rooms. So ISO 8 is grade D, and the operations that are done in grade D are vial decartoning, equipment cleaning, entry airlock to grade C, primary gowning area or first gowning area from corridor to the compounding area. So corridor is a CNC, right? Control not classified, and packaging areas are typically control not classified. When you enter from control not classified area to a grade C area, you need an airlock or you need a room that is between C and C to grade C. That is the room between those C and C to grade D is, sorry, C and C to grade C is a grade D, because you can't skip two classifications. You have to go from one classification above. So you can go from C and C to grade D, and from grade D to grade C you can go. You can't go directly from C and C to grade C. Uh, that's not acceptable. Some references here, as you can see, ISO 146441 standard and FDA guidance for industry, sterile drugs produced by aseptic processing, and Annex 1. Good manufacturing practices guide, manufacture of sterile goods, sterile drugs, and Annex 1, manufacture of sterile medicinal products. So if you have any questions, drop me in the comments, and if you have any video suggestions, please drop me. And if you think this is helpful, do let me know, like, share, and subscribe. Um, thank you.